In this lesson, we're going to be focusing on prokaryotic structures. So if you remember from our previous lesson, there are two different types of cells in biology, prokaryotic cells and eukaryotic cells. This lesson will focus exclusively on prokaryotic cells. Specifically, we're focusing on bacterial cells. And our next lesson, which will be the eukaryotic cell lesson, will focus on things like fungi and protozoan, which are eukaryotic cells that are also considered microorganisms. So one of the first things we need to discuss are the primary differences between prokaryotic and eukaryotic cells. So one of the primary differences is the way that these cells package their DNA. So with eukaryotic cells, they traditionally package their DNA inside of a membrane-bound nucleus. This is probably what you're most familiar with, having gone through other biology courses. When you talk about eukaryotic cells, such as human cells and other animal cells, they do have a nucleus with chromosomes or packaged DNA within. But with bacteria, which are prokaryotic cells, they lack a membrane-bound nucleus. They do have a chromosome, they do have genetic material. It's just not encased in a membrane-bound organelle, such as what we'd expect for a eukaryotic cell. The second difference has to do with the composition of the cell wall. Both prokaryotic and eukaryotic cells can have a cell wall, but they are composed of different things. So with prokaryotic or bacterial cells, their cell wall is composed of a chemical called peptidoglycan, and that chemical is exclusive to bacteria. It is only found in bacterial cell walls. If a eukaryotic cell has a cell wall, it will not contain peptidoglycan. Not all eukaryotic cells have a cell wall. For example, humans have eukaryotic cells, but we lack a cell wall. But things like fungi and plants, they're eukaryotic cells. They can have a cell wall, but that cell wall will not contain peptidoglycan. It will often contain something like chitin or cellulose, but not peptidoglycan. Again, remember, peptidoglycan is exclusive to only bacterial cell walls. And the last difference are internal structures. So with eukaryotic cells, they have all the standard internal structures that you're likely familiar with. The nucleus is one, but then also things like the Golgi apparatus and the endoplasmic reticulum and mitochondria. Whereas prokaryotic cells, such as bacteria, they lack all membrane-bound organelles. They really are just composed internally of cytoplasm, some genetic material, and typically ribosomes for protein synthesis, but they lack any type of membrane-bound organelles. So these are the various prokaryotic structures that we're going to be going over in this lesson. These are the structures that are found in all bacterial cells. So all bacterial cells need these structures in order for kind of baseline survival. And then these structures over here are the more advantageous structures. So some bacteria have them, some bacteria don't. Those bacteria that have these structures typically have some type of an advantage. But we'll talk about all these structures throughout the course of this lesson. Here's a basic diagram of a simple bacterial cell. And if we kind of compare this mentally to what we've learned with other cells, typically eukaryotic cells, it's much less complex, especially internally. So we don't see all the traditional membrane-bound organelles. Again, the Golgi apparatus, the nucleus, the endoplasmic reticulum, the lysosomes, the vacuoles, the mitochondria, they're all absent in prokaryotic cells. Prokaryotic or bacterial cells really just have they have their genetic chromosomes, their DNA, cytoplasm, and then some ribosomes internally. Now they can have quite a few external appendages and layers that we'll talk about, but internally they're not nearly as complex as what we're used to with eukaryotic cell structures. And we're going to take the time to go over each of these structures. So with bacteria, they are independent single-celled microorganisms, which is different from humans. I mean, as humans, we are obviously macroscopic. We can be seen with the naked eye. And as an organism, we are composed of billions of cells. Whereas with bacteria, they're microscopic, so they need to be seen with magnification, and they are individual cellular organisms. The two most common shapes that bacteria can exist in are cocci, which we learned in an earlier lesson means circular shape. These typically have a circumference of about 1 micrometer. Then there are the rods, which we often call the bacillus bacterium. They are double the size of the coccus bacterium and are about 2 micrometers in length, 1 micrometer in width. Some bacteria specifically stick to their original shape, but there are bacteria that can change their shape. Those bacteria that are able to change their shape are said to be able to go through something called pleomorphism. So pleomorphism is when bacteria can change their size, shape, and even potentially arrangement in response to either a genetic mutation that they've acquired in their DNA or some type of change in the environment, such as access to nutrients. So if a bacterium exists naturally in the environment as a coccus, and we try to grow it in the lab, which is kind of obviously an artificial setting where the nutrients might be different, there is the potential for the bacteria to go through pleomorphism and change its appearance as far as things like size, shape, and arrangement. So anytime we grow bacteria in the lab, we want to mimic the natural environment as much as possible to prevent pleomorphism if we're trying to study what bacteria look like in their natural environment. So some of these bacterial shapes we've already covered in previous lesson, and some are new. So we do know that bacteria that are coccus in shape are going to be circular, and that bacteria that are rod-shaped are going to be bacillus. Now there is a hybrid, coccobacillus, which are basically very short rods to describe them best. They are more of kind of elongated ovals, so they're not perfectly circular like what we would expect for coccus, but they're not as long as a typical bacillus or rod-shaped bacteria. So they really are just a hybrid between coccus and bacillus. Then there's Vibrio, which are curved rods. And then we did mention in a previous lesson Spirillum, which Spirillum are the kind of loose, wavy type bacteria. One we haven't talked about yet are spirochetes, and I do realize that these look very similar here, but a spirochete bacterium is going to have more of a helical type appearance. So imagine like a slinky. That's kind of what we would expect for a spirochete type bacteria, to have more of a helical appearance, whereas Spirillum are simply just a loose wave. So now that we know the various shapes of bacteria, we're going to focus on arrangements. With arrangements, we'll primarily focus on coccus and bacillus shapes. Most of the Vibrios and the Spirillum and the spirochetes, they typically exist as single cells, so they don't often form various arrangements. But the coccus bacteria, which are the circular bacteria, and the bacillus bacteria, which are the rod-shaped bacteria, they are prone to having a variety of arrangements. And how a bacteria gets its arrangement is based on how they stay connected to one another after a division cycle. So when bacterial cells divide, they always double themselves. So 1 will always become 2, and then those 2 will divide and become 4, etc. Sometimes they completely separate after they go through a division cycle, but then sometimes they stay stuck together in the form of different arrangements. So coccus bacteria definitely can make the most diverse arrangements. So if it's just a single coccus, so if a coccus bacterium goes through a division cycle and forms 2 coccus bacterium, and they completely separate from each other, then those coccus are said to have monococcus arrangement. So they're just a single circular bacterial cell. If they divide and stay stuck together in a pair of two, we say that's diplococcus. So diplo for pair, and then coccus for being circular. If they continue to divide and stay attached to form a chain, then we classify that as streptococcus. Strepto meaning chain, coccus again meaning circular. Some coccus bacteria can attach in a square formation, so it would just be 4 bacterial cells forming a square, 4 circular bacterial cells forming a square. We often call this arrangement a tetrad. The most common is definitely Staphylococcus. Staphylo just means irregular grape-like clusters. So when we see Staphylococcus, we're just thinking clusters of round bacteria. Sarkinia it's very similar to tetrad, however it's a 3D version of a tetrad. So it's kind of hard to see in this picture, but picture this as a 3D cube, whereas this is just a tetrad's just on a single plane, so it would just be 4 cells stuck together. Over here we would see more of a 3D cube formation of the coccus cells arranged together. Now with bacilli or the rod-shaped bacteria, they can form a variety of arrangements too, not nearly as many as the coccus-shaped bacteria. Their arrangements typically have to be in some type of linear format, so they have to be arranged in some type of line. We already mentioned that there is kind of this hybrid coccus bacillus, which is more of a very short rod or elongated coccus, so it's kind of a combination of the two. With the rod-shaped bacteria, they can just have single cell arrangement. So here we see a bacilli that's just by itself. So it just kind of has that singular arrangement. Just like Streptococcus, bacilli can form pairs of 2. So here we have a diplobacilli, so a pair of 2 bacilli. They can also form chains. So here we see a chain of bacilli, so this would be a streptobacilli. Palisades are basically when the bacillus-shaped bacteria form a kind of stacking arrangement where they are connected to each other in a vertical formation, such as what we see here. So it's more of a vertical stacking is what we'd expect for palisades. Bacillus bacteria are not able to form any type of irregular clusters. So if you ever see bacillus bacteria that appear to be clusters, it's probably just where quite a few of them are confined to a small space and they're not actually attached to one another. We see that quite often, but they typically do have to form some type of linear arrangement. So we'll first kind of go through and talk about the external structures that bacterial cells can have. There are various appendages that bacterial cells can have, one being flagella that they use for movement, fimbriae that they use to attach to surfaces such as cells and tissues in our body, and then pili that they can use to attach to other bacterial cells for the purpose of communication.